So first of all, a bit about the neuro this semester. It's not as bad as the anatomy. Okay, so anatomy there you had lots of information. It was anatomy, histo, embryo. It was insane there. Neuro is a bit easier. Okay, a bit easier because the amount of material is definitely less. The problem with neuro is that there are some things which are just entire memorization, especially if it comes for the anatomy part. So there's no understanding whatsoever. You just memorize and that's it. That's how you should be there. For histo and embryo, you can actually understand a couple of things, but for the anatomy part, just make sure that you don't leave it behind for long. Make sure that once a week you actually do revise just a bit, because by the time you reach the final, it will definitely be like a lot. In this course, of course, our main focus is histo and embryo. As I told you, it's going to be 8 lessons, but I do want to incorporate just a bit of the anatomy, the one that I think will be very useful for our course right now. So let's take a look. What do we have here? Do you know how the final exam will be? No. Okay, so your final exam will be that you have neuroanatomy exam. So you will choose 2 topics. One of them is brain. Another topic is spinal cord, ear, eyes, I don't know, all of this crap. It's an ID topics, right? So you have to show things with a bit of a theory. And there's also going to be a written exam. Written exam consists of 60 questions. Written exam consists of neuroembryology, neurohistology, neurophysiology, and a bit of the neurobiology. Neurobiology are just minimals. I will send you the list. It's just like, I don't know, like 2 or 3 pages of minimals. You memorize them and that's it. For neurophysiology, the only source that you need is actually the physio lectures. So make sure that whenever it actually says neurophysio, you go and attend that lecture because that's where the questions will be from. Neurohisto and neuroembryo, I am here for you. At the end of this course, there's like a huge file made by me and a couple of students. It consists of more than 300 past papers questions. It's some of them are solved, some of them are unsolved, but it's a good thing to go over that. Okay. Overall, it's not a difficult subject. Like, trust me, like it's not difficult. It's not the same as anatomy. So as long as you are studying on time, everything is going to be great. And we are studying on time. Okay, so
is it going to be MCQs? The 60 questions?
Sorry?
Is it MCQs?
Yeah. It's the same. It's very similar to like your physio situation, you know? Sometimes there is a key, sometimes there is no key, you know, like this kind of stuff. So yeah. But trust me, it just kind of sounds bad, but it actually isn't. Like it isn't so bad.
Do we have SCTs also?
No, you have no SCTs whatsoever. So basically, you're preparing for the final exam.
Okay.
Mm-hmm. So that's why lots of students, because you know you have physio SCTs and you have biochem SCTs. That's the problem. Like throughout the semester, people focus on physio and biochem and leave neuro for the last thing. But it's very easy to, like physio in the final, very easy. Biochem in the final, very easy. Neuro is the one that will like tickle your nerves just a bit. So that's why I always say just go first week, just get rid of neuro and you will feel much better that you're done with the anatomy department forever. Like that's it, you know, so no more. But yeah. Okay, so with that being said, let's take a look. What do we have here? So this is very, very much made easy for you guys. So first thing first, for sure, you know that we have 2 types of nervous systems. We have the central nervous system and we have the peripheral nervous system. This is like Neuro 101. What do we have within the central nervous system? Within the central nervous system, we have the cerebrum, right? So that's your brain, just a fancy way of saying brain. We say cerebrum. A bit below the brain, we can see this area right here, which is going to consist of brain stem. it's also part of your CNS, and cerebellum, which also is part of your CNS. And a bit downwards, you see the spinal cord. So these four structures, they make up central nervous system: cerebrum, cerebellum, brainstem, and spinal cord. In return, you can see that brainstem also consists of three parts. So midbrain, pons, and medulla oblongata, right? So you can see all three of them are located at the level of the cerebellum. Within your cerebrum, there is a super important structures there that we call diencephalon and basal ganglia. You can see that they're sitting sort of in the center of your brain, right? Diencephalon, it just consists of a bunch of scary sounding structures. Like the scary sounding structures are all finishing with the word thalamus. So whatever you can come up with, if it ends with thalamus, it belongs to the diencephalon. So for example, epithalamus, hypothalamus, right? Subthalamus, thalamus itself, it will all be the structures which belong to the diencephalon. Basal ganglia. Anybody knows what is ganglia? What is this word right here?
A group of nerve cells.
Very good. It's like exactly. It's like a group of cell bodies, right? So we will talk about it right now. But it's a group of cell bodies of neurons. Can ganglion be in the central nervous system, or is it only in the peripheral?
No,
It can be in
it
both.
can be in the CNS. It can be in both.
Actually, the word ganglion itself is meant only for the peripheral nervous system. So the word itself, ganglion, you can never say that there is ganglion in the central nervous system. That's a fatal mistake. The word ganglion itself, this collection of neurons, it's typically reserved for the PNS. But this is the exception right here. This is the exception right here. Technically, it's not really ganglion. How do we call collection of the cell bodies in the central nervous system?
Nucleus.
Yes.
Very good. You call it nucleus. So technically, you should call it basal nuclei, but nobody calls it like that. So they just call it basal ganglia. So you should know that this is a very interesting exception right here. I repeat, the word ganglion and the word nucleus, they are talking about the same thing. You will see that the problem with neuroanatomy is that there are so many terms for the same structure. There are so many different words for exactly the same structure. But here there is a bit of a like difference between them, right? So I repeat that ganglia are collection of cell bodies in the peripheral nervous system, while nucleus is in the central nervous system. Basal ganglia are in the central nervous system. They're within the brain. So which kind of nuclei are in the basal ganglia? You can see them here. That's not all of them. There are a bit more. It's just the most important ones. So for example, caudate nucleus, globus pallidus, there are actually two of them. Globus pallidus externa, globus pallidus interna. So both of them are part of the basal ganglia. The putamen, claustrum, and amygdala. Okay, there's additional one, which is the red nucleus and the substantia nigra. They also belong to the basal ganglia as well, but we'll talk more details about it as we go. Okay, so this is just like some kind of overview for you guys. Yeah. We have to know, we will see for this course that we will talk details about diencephalon and basal ganglia, but surprisingly, they are not connected. There is one connection and that's it. So these two structures, as you can see, are sitting within the core of the brain. But surprisingly, the two areas do not intersect much. So it's like they're just neighbors, but they do different things completely. So the two structures have just one intersection, and we will talk about which kind of intersection specifically. Okay, what is the basic functional unit in the central nervous system? Of course, it's going to be a neuron. For sure, you know how the neuron looks like, right? So this is like a schematic representation. We have the cell body, we have a bunch of, how do we call this?
Dendrites.
Dendrites, and we have
Axon.
axon. What is synapse? How would you describe it?
A junction between the presynaptic and postsynaptic neurons.
Very good. Yeah. So it's some kind of junction between the presynaptic and postsynaptic. The better word that I would use is actually communication, because that's exactly what is happening at this level. The two neurons are communicating somehow. So anatomy department will also appreciate it if you would say that synapse is a communication, or as Simran said, junction between the two neurons, pre and post, right? But again, we'll talk more details about it. For sure, you know that how exactly do they communicate, right? So there is an action potential, which is just propagating in a certain manner, right? So from one neuron to another, it keeps on jumping this way. Now, a chain of communicating neurons is called pathway. And when we will talk about spinal cord, you will see that inside of it, there's so many pathways. Okay, there's so many. And unfortunately, we'll have to know what are their functions, where do they begin, and where do they end. But generally speaking, the area, like a chain of neurons, one after another, right? So you're going to call it as a pathway. Within the central nervous system, a bunch of axons together, right? You can imagine that there are many neurons sitting next to each other. A bunch of axons together in the central nervous system is called tract, fasciculus, peduncle, or lemniscus. 4 names for the same thing. So whenever you hear these names, what are you thinking about? You're thinking about a bunch of axons together, just lots and lots of axons sitting together. How would you call exactly the same structure in the peripheral nervous system? It's simply a nerve. So any nerve that you can imagine, it's the same thing, right? So again, location matters. In the central nervous system, a bunch of neurons, axons together have different names: tracts, fasciculus, peduncle, lemniscus, etc. In the peripheral nervous system, you will simply call it a nerve. Yeah. Okay, speaking of nerves, you know that we have different types of nerves. We have the spinal and the cranial nerves, right? So those are the typically, of course, there's some additional nerves, but those are the big ones, the spinal and the cranial nerves. There are 12 cranial nerves, and there are 31 pairs of the spinal nerves. And you remember that they have a very interesting arrangement, right? So how many cervical nerves do we have?
8.
Very good. We have 8 of them. How many vertebrae do we have in the cervical region?
7.
7.
So where is the 8th? Where is the C8?
Underneath
nerve?
the 7.
Very good. It's underneath the 7. So you should take a look at it this way. Okay, so this is the most important part, that C8, it's somewhere underneath the 7th. Then where is the C1? Right? As you can see, it is above the C1 vertebrae. So you can imagine that there are 7 vertebrae, right? But if you count the nerves, there are 8 of them, because the first one does not begin under the vertebrae, it begins above it, right? So C1, 2, 3, 4, 5, 6, 7, and then 8th is under the C7. Thoracic continue, right? So there are going to be 12 of them. You can see also below, right? And etc. So for sure, you know that the spinal cord itself is much shorter than the vertebral column, right? So where does the spinal cord finish? Typically, it finishes at the level of L2. In the book, they say L1, L2, but from clinical practice, it's L2 typically. Then from the L2, you can see that there are a bunch of nerves here, bunch of them, right? So a bunch of spinal nerves are just sitting here below the L2. Which nerves are these? These are the L2 to S5 nerves. And because they're going to much deeper level, as you can see, they're elongating, right? And this creates this horse tail appearance. So this end of the spinal cord, which consists of L2 to S5 spinal nerves, has a name in Latin language. It's called cauda equina. Okay, so cauda equina or horsetail. Okay, that's my fault here. I apologize. Just a sec. Discard. Okay. I don't know why did that happen, but just a sec. Okay, share like this. In your slides, is it also like this? Or is it, how is it there? Is it cut in half or something? Huh, guys?
It is cut in half, but I think we can see the entire image. I mean, the two of
Because like
images.
this image is here, right? You can see it on the next one. Yeah, so I'm not sure why is it actually here, this part, but it's okay. Anyway, so most important things is that here we will see that they're going to be the cerebrum itself, right? So the brain itself. So you know that it consists of different lobes. So we have the frontal, parietal, temporal, and occipital lobe, of course. How do you call these deepenings? Like, do you see there are some kind of deepenings into the brain? You will call them sulci. Okay, so you will call them sulci. Gyra are the protrusions itself, right? So you can imagine that if I actually draw it, draw. Okay, if you can imagine, if I draw it, this is gyrus number 1, this is gyrus number 2, gyrus number 3. I'm talking about these protrusions of the brain, right? These gyri, you will see that they have all the different names. That's the first lesson in the neuroanatomy, to know how do you call this gyrus, how do you call this gyrus, and etc. So each and one of them has a very particular name, so you must be able to recognize them. But how do we call the deepenings between the gyri? We call them sulcus, right? So this is one sulcus or many sulci. Most important is definitely the central sulcus and the lateral sulcus. I know it's written here fissure, but it's actually a sulcus. And very important to know the people's names. So you need to know that central sulcus is the Rolando's sulcus. This dude will come up like quite often. You will see him quite often in your anatomy. The lateral sulcus is sulcus of Sylvius. Also, you will see him quite often. There are many things that are named after them. Fun fact, it's actually an embryological fun fact. When do we start to get the gyri as an embryo?
Week 3.
Week 3, it's a bit too early because there's, as you remember, week 3, we're just starting to make the neural tube, right? The whole process, neurulation, we're just starting to make the brain and spinal cord. It's actually as early as 4th month. So approximately at starting from 4th month of embryological development, this is when these particular gyri are starting to appear. Before that, the brain is smooth, completely smooth. There is nothing of a matter like that, right? Okay, and one more fun fact. The more you learn, so we are in med school, the more we learn, the more curvy the gyri become. So that's why whenever you're done with life, if you actually look at your brain, if you had the ability to look at your brain as after med school or after like teaching or constantly learning something new, you will see that your brain is very like very, very wavy because these gyri are just protruding everywhere. Okay, so this is going to be exactly the same thing, just from different perspective, right? So this part right here. So this is your brain right here. This is the diencephalon, right? Here we have a bit of the basal ganglia as well. And this is the brain stem that is already starting in cerebellum, right? And etc. I want to get your attention to this part right here. There's something which is called clivus. Do you see this? Clivus? They will ask you about it in the anatomy. Clivus is a piece of bone, and this is a very, very thin bone, to be more specific. And the problem with clivus is that tumors which are originating in your nasal cavity easily protrude through clivus and kind of inseminate your brainstem. So that's why if you have some kind of nasal or nasopharyngeal tumors, they can easily spread to the central nervous system, all the way to the brainstem, and then from the brainstem all the way to the cerebrum itself. Why? Because of clivus. Because clivus is a very, very, very thin bone. Okay, so, and another thing that they can ask you here, which of the structures of the brain stem is sort of pushed against clivus? That would be pons. I know in this picture it's not very easily seen here, but that would actually be pons. So pons is the one which is pushed against clivus. So hypothetically, if you had a nasopharyngeal tumor, the first thing that it was spread to would be
Pons.
Pons.
to be pons. Yeah. Okay. Okay. So that's a bit of the neuro anatomy right here. So just a sec. Cancel. Okay. So let's go for the neurohisto. Here we begin with a little bit of the functional part because it's quite important for us to know, obviously. So we're going back to the idea that we have, anatomically speaking, we have two nervous systems, right? We have the central and peripheral nervous system. We have named which structures belong to the central nervous system, but which structures belong to the peripheral nervous system? What could you say? Where is peripheral nervous system?
Anything that's not brain and spinal cord.
Exactly. It's everything else. It's anything that is not the central nervous system, that would be the peripheral nervous system. So for example, cranial, spinal, peripheral nerves, right? So these are definitely the peripheral nervous system. And of course, the ganglions. Ganglions, we have said, are a bunch of cell bodies outside of central nervous system. They belong to the peripheral nervous system. Again, tell me, how do we call the same thing in the CNS?
Nucleus.
Very good. We'll call it nucleus, so don't forget about that. Nuclei belong to the CNS. Ganglions belong to the PNS. That's anatomical division of the nervous system, but there's also functional division of the nervous system. Functionally speaking, nervous system is divided into the somatic and autonomic nervous system. Autonomic nervous system is further subdivided into the sympathetic and parasympathetic divisions. What is the major difference between autonomic and somatic?
The voluntary one is voluntary.
Exactly. So which one is voluntary?
Somatic.
Somatic. Somatic is the voluntary part of your nervous system. So whenever you're thinking about voluntary control, forget the sympathetic, forget the parasympathetic, forget any of those things. You're talking about somatic nervous system, to be more specific, right? Somatic nervous system will have two components to it. That will be the sensory and motor component. That's like its subdivision. Autonomic is everything which is involuntary, and we have said sympathetic and parasympathetic. So a bit more about somatic nervous system. We have said that it's two its two subdivisions, if we could say so, would be the sensory and the motor, right? Another name for sensory nerves, they're also called afferent nerves. Another name for motor nerves, they're also called efferent nerves. Try to answer why. Why is this afferent and why is this efferent?
Sensory nerves will bring the sensation like to the brain. So it's like afferent is like towards the brain.
Exactly. The
Afferent is
main
away from.
Exactly. The main player here is definitely the brain. So sensory neurons, sensory nerves, they're bringing the information to the brain. So afferent, right? So that's what they're doing. I'm touching the table right now. Right now, all of my Pacinian corpuscles in my hands are actually going ballistic. Okay, so they're like, yeah, that's definitely the brain. That's definitely the table. I can feel the pressure. So all of the Pacinian corpuscles are firing action potential, which are traveling along the sensory nerves and going all the way to my brain. So of course it makes sense that sensory are afferent to the brain. Maybe this table is a bit not to my liking. Maybe it's a bit cold. I don't want to touch it anymore. So my brain does not like it. So what should it do? It should send the action potential to my muscles so I can remove my hands from said table. So which nerves are going to do so? Of course, the motor nerves to the muscles. So what is their name? Efferent nerve. So a question, can motor nerve ever be afferent?
No.
Absolutely not. Yes. So that's going to be a very interesting option in your exam, in your written exam. Okay, so read the question very carefully. So they will say motor nerve and then brackets afferent nerve. That's a mistake right there. Okay, so it's not afferent. Motor can never be afferent. Motor is always efferent. Sensory is always afferent. Make sure you read the question correctly. Okay, we have said that somatic nervous system has two subdivisions, right? So the motor, the sensory, which nerves specifically would be divided into sensory, motor, the spinal and the cranial nerve. I forgot to mention a very important part here. Speaking of cranial, we have said that cranial nerves belong to the peripheral nervous system. All of them but two. Which two? Which two cranial nerves do not belong to the peripheral and instead belong to the central? Try to think, anatomy.
Cranial accessory.
Again?
Cranial accessory nerve.
Nerve number
Accessory
one.
is somewhere here, it's a bit not there. It's actually quite easy. It's going to be olfactory and optic. These are the two cranial nerves which actually belong to the central nervous system and not to the peripheral nervous system. Why is that? Because olfactory and optic nerves, you will learn it in embryology, they are actually direct extension of diencephalon. You remember diencephalon? Everything with the thalamuses, epi, sub, whatever, right? So the optic and olfactory nerves are actually embryologically considered to be direct extensions of that. So that's why they actually belong to the central nervous system. Okay, nerve tissue. Nerve tissue has two principal types of cells. We have the neurons and the supporting cells. For sure, you know, how do we call supporting cells?
Glial cells.
We call them glial cells. I'll be saying glial, but yeah, technically it's glial, but I'm used to saying glial, so glial cells, okay? So glial cells are the supporting cells. Fun fact, in our university, technically, glial cells are supporting cells of the central nervous system only. So you cannot say that glial cell is a supporting cell of the PNS. Okay, it would be a bit incorrect. I think they will change the stupid rule this semester, but last year that was the case. Okay, so you would have to say that glial cells are only found in the central nervous system. Yeah. Okay, let's take a look at anatomy of a neuron. You remember that functional unit of the nervous system, this is going to be a neuron. We have said that neuron consists of the cell body, dendrites, and an axon. What can you find within the cell body? What is inside there?
Nucleus.
What else?
Rough endoplasmic.
Mitochondria.
Mitochondria, I heard. What? Somebody else was saying something, what?
Rough endoplasmic reticulum.
Oh, sort of endoplasmic reticulum, very good. Anything else?
mTOR.
Cytoplasm.
Cytoplasm, what else?
Microtubule organizing centers.
Microtubule organizing center, very good. Anything else? You see where I'm going with it or no?
No.
No. Anything that you can find in any other cell would be here. So whatever organelles you can come up with, they'll be there. They'll be in the cell body. So technically, any other cell, like, I don't know, epithelial cell, I don't know, muscle cell, right? So whatever it has, it will be right here in the cell body of the neuron. Yeah. Okay, so all of the particular organelles, etc. Cell body has another name, actually 3 names. So number 1 name is a cell body. Second name is soma with the S. Okay, so soma, that's another name for cell body. And last one is perikaryon. That's the one that you might see in the exam. So either cell body or perikaryon. So perikaryon, it is the cell body. Remember I told you neuroanatomy, lots of names for the same thing? It's a big issue actually, but yeah. Now, dendrites here, they are some kind of cytoplasmic protrusions of the cell body, right? So just like feed processes of the cell body. What is the job of dendrites? What do they do?
Synapse.
Okay, if there's another neuron, sure, but say better.
They receive the impulse.
Very good. The keyword that you're going to go with is receiving. So these dendrites, they are the receiving side of a neuron. They receive the impulse of some sort, whatever this impulse may be. What do you think the function of the axon is? Keyword.
Transmission of an impulse.
Transmission, yes. Receive, transmit. Receive, transmit. Can an axon receive?
Yes.
We said that dendrites are the receiving side, but can an axon receive?
Yes.
Yes. How would we call that synapse? We will call it axonic. So imagine there's another axon that is coming in this way like this. Okay, so that's going to be a synapse right here, and we'll call it axonic synapse. And technically, hypothetically, this part, the second axon, it is receiving. So technically, theoretically, hypothetically, whatever you want to say, it could receive. Yeah, axon could receive. Can dendrites transmit?
No.
If axon can receive, what's stopping dendrites from transmitting? Nothing. So in theory, there is another synapse, which is called dendritic. So axonic, dendritic. And in theory, it can transmit. Which one do we see in our body as humans? We can see axonic, but we almost never, almost impossible to find dendritic. But again, in theory, it could. In theory, it could. Just in reality, on practice, you probably won't find one. Okay, so we have talked about axons, we have talked about dendrites, perikaryon, right? Soma, that's the whole thing. Okay, how do we call these things? This is the myelin sheath, right? In the peripheral nervous system, who is responsible for giving this myelin sheath?
Schwann
cells.
cells.
Schwann cell. Where does the Schwann cell come from?
Neural crest.
Neural crest cells, very good. So Schwann cells arise from the neural crest cells. Can we have Schwann cells in the central nervous system?
They would be the oligodendroglaya, right?
Amazing. They will not be Schwann cells, so we cannot have Schwann cells in the central nervous system. In the central nervous system, there is a cousin of the Schwann cell, which is called oligodendrocyte. Yes. Okay, so oligodendrocyte. They have the same job, which is to produce myelin sheath. Oligodendrocyte makes myelin sheath over the axon in the central nervous system, and Schwann cell does the same thing in the peripheral nervous system. What is the purpose of the myelin sheath? Why do we need it?
Helps in saltatory conduction, so transmission of impulses faster, and also gives isolation.
Insulation, isolation, saltatory conduction makes faster. How exactly?
The myelin sheath is found in like spaces in places, so it makes like nodes of Ranvier.
So these gaps right here, right? So these
Uh,
spaces between the myelin sheaths, as you have said, they have a name. They're called node of Ranvier. Yeah. But how exactly the myelin sheath helps the faster propagation of action potential?
Doesn't
No action
allow
potential.
conduction in that area.
Again, because two of you were talking.
Doesn't allow conduction in that the area where the myelin sheath is present.
Okay, so, but
Then
how is it still going
distance.
this way? Why is it going there? You're absolutely right. The only area where we can actually add a bit more of the action potential will be in the nodes of Ranvier. That's where we could fire it up. But what makes sure that action potential goes, it actually travels that way? The myelin sheath. That's what is responsible for that. My question is, how exactly? How exactly is myelin sheath allows the action potential to go in that direction? When we say that,
diffusion.
sorry?
It allows diffusion.
So close. Okay, so when we say that myelin sheath is insulating, what do we mean by that? What is it insulating from?
Charges
So what is it not allowing charges to do?
to accumulate.
Create an action potential.
It does not allow charges to leak out. It doesn't allow them to leak out. If the charges are kept within the axon, that means that action potential can go. It can actually travel. It's like you're insulating your windows to make sure that the warm air does not leak out, that it stays within the room. The same idea is here. Myelin sheath, I'm not talking about nodes of Ranvier. Nodes of Ranvier are empty spaces. That's where I can add more ions, I can add more action potential, etc. How exactly myelin sheath makes sure that action potential propagates? The idea is that myelin sheath insulates the axon. It fully coats it like that. So whatever ions, whatever charges you have in the axon, they simply cannot go anywhere. They have to go straight, right? So they have to propagate forward. So that's the idea there. Okay, so we have talked about this part. Okay, this question, axon hillock. What is that?
Where you can't find a lot of organelles.
So, so that's axon hillock is this area right here. Axon hillock is the area where axon will begin, right? So it's like It's like a border between the cell body of a neuron and the axon itself, right? So that's like kind of an overview. Now, within the axon hillock, we have here some kind of segment. How do we call that segment? That's where the action potential begins, right here. Within the axon hillock, there is a part there which is free of myelin sheath, of course, and it is called initiation segment. Initiation segment, that's the area where action potential fires up. That's the area where the ions do the movement that creates the action potential. Okay, so it's going to be right here. Okay, you remember we talked about it, fascicles, tracts, right? So it's just a bunch of axons together. We talked about it, so just read it again. Okay, let's talk about the supporting cells. The supporting cells are the other type of a cell in the nervous system, and most importantly is that they're non-conducting cells. They absolutely cannot at any time, never ever, conduct and propagate the action potential, right? So they are simply supporting cells. Do not forget that we have two nervous systems, CNS and peripheral nervous system. In the central nervous system, the supporting cells are going to be called glial cells or neuroglial cells. I truly hope that this semester they change it and allow you to say that in the PNS we also have neuroglia. But for now, make sure you check whatever they wrote in the lectures or seminars. For now, neuroglia are supporting cells in the CNS specifically. Which supporting cells are in the PNS? Well, of course, the Schwann cell and the satellite cells. Schwann cells in the PNS, right? Schwann cells in the PNS surround axon. You can see many Schwann cells surround one axon. Satellite cells in the PNS surround cell body. You will see them as the simple cuboidal cells, which are simply insulating the cell body, right? So Schwann cells insulate the axon in the peripheral nervous system. Satellite cells are going to be insulating the cell body. They are supporting cells. So as always, green light to bullshit, whatever you can come up with, they probably do it. Okay, so physical, emotional, whatever, electrical, metabolic exchange, whatever you can come up with. Now let's take a look at the central nervous system. In the central nervous system, we have 4 types of neuroglial cells, 4 of them. Oligodendrocytes, astrocytes, ependymal, and microglial cells. We already said something about oligodendrocyte, right? What was oligodendrocyte doing?
Making the myelin sheath in the CNS.
In the central nervous system, yeah. So which cells are oligodendrocytes for us? The blue ones, okay? So this cell, this is the oligodendrocyte right here. What is the one thing you notice compared to Schwann cell, oligodendrocyte? What do you notice?
The nucleus is not around the axon.
So you can see that the cell is not around the axon, its cell processes are. So, Schwann cell. How many Schwann cells surround one axon in the PNS? God knows, but it's many Schwann cells per one axon. What about in the central nervous system? Oligodendrocyte, what do you see? Minimum 2 axons are being insulated by oligodendrocyte. I like to think that oligodendrocyte is a very promiscuous cell, okay? So it's a very, in simple English, slutty cell, okay? It just goes around, man. It just goes around in the central nervous system, myelinating as many axons as it possibly can, okay? So compared to a Schwann cell is a very, very faithful kind of cell because all it does is just simply insulating one axon. You can see there are many Schwann cells for one axon in the peripheral nervous system, but there are many many axons per one oligodendrocyte in the central nervous system. Okay, let's talk about microglial cells. Microglial cells are macrophages of the central nervous system. So they are the phagocytotic cells. All they do is just, I don't know, phagocytose some pathogens, remove some dead cells, blah, blah, blah. So microglial cells are phagocytotic cells. They're going to be this one. Okay, so that's the one right here. This is the microglial cell. Okay, next cell is going to be astrocytes. We will talk about it that there are two types of astrocytes, fibrous and protoplasmic astrocytes, the two different types of astrocytes, fibrous and protoplasmic. As you can see, the astrocyte, what is it doing? It's kind of connecting the neuron to a blood vessel, right? It's simultaneously hugging the blood vessel and the neuron. So what do you think? What could be one of the functions of astrocytes?
Nutrition.
Nutrition.
Or nutrition. Anything else? It surrounds a blood vessel.
So maybe sensory.
Blood pressure.
Blood-brain barrier. Okay, so it creates a barrier of some sort. It's additional layer that things from the blood have to pass through in order to get to the neuron, right? So you're going to see that astrocytes, they are quite important because they actually participate in the blood-brain barrier. Okay, ependymal cells. Ependymal cells are these ones on the top, okay, that you can see here. Without going too much details, inside of
what is exact?
Sorry?
What is it on top of? What is the entire thing?
This is the ependymal cell, and these things on the top are cilia.
No, I'm talking about, you said they're on the top of something. I didn't get it.
Oh yeah, so I'll explain it now. Okay, so they're on the top of this thing, so I'll explain it now. So without going too much details, what exactly they do and what exactly they like kind of don't do. Inside of your central nervous system, you have lots and lots of cavities. Like inside of your brain, you have a cavity. Inside of your midbrain, you have a cavity. Even in the spinal cord itself, there's also a cavity. And maybe you know, what is this cavity filled with?
Cerebrospinal fluid.
Exactly. It is filled with cerebrospinal fluid. Who makes this cerebrospinal fluid?
Ependymal cells.
Ependymal cells. Ependymal cells are responsible for creating this cerebrospinal fluid. So this, which you can see here, this is the cavity that I'm talking about. So this is like a piece of brain, for example, and this could be, for example, like a lateral ventricle. We will learn exactly where these cavities are, but they are hollow spaces, and these spaces are filled with some kind of fluid, which we call cerebrospinal fluid, of course. And these cavities are lined by some kind of cells. Do you see they're lining this cavity? So these cells, they're called ependymal cells, and their job is to create CSF. What do you think they use as a base for CSF? What are they making CSF from?
Blood plasma.
Very good. From the blood plasma, very good. You can see that they're also near a blood vessel. So these ependymal cells, they're going to be basically like sucking up all of the blood plasma, removing the proteins, and converting it into the CSF. Again, we'll talk more details about it. But I do want to add it right now that there is something, okay, so ependymal cells plus blood vessel plus has a name. It's called choroid plexus. So typically in these cavities inside of your brain, inside of your spinal cord, you will see that there is a bit of a tissue. It's called choroid plexus. choroid plexus is ependymal cells plus capillaries. So a bunch of capillaries on which ependymal cells are sitting. So in a way, if we actually have to be very specific, right? What exactly produces the CSF? You're right to say ependymal cells, but you'll be more correct to say that what exactly makes CSF?
Choroid plexus.
Choroid plexus. And I repeat, what is choroid plexus exactly?
Ependymal cells plus blood vessel.
Plus blood vessels, yeah. Okay, here are the functions. We talked about them.
Julia,
Mm.
do we see ependymal cells last semester in the pineal gland?
In the pineal gland, we could have, because it has there is a bit of a ventricle there. There is a bit of a cavity there, so we could have seen, yes. So some professors could have shown you. I didn't, for sure I didn't, but some professors could have possibly done it, yeah, because the pineal gland is right next to the lateral ventricle, so for sure, yeah, for sure. Okay, so you remember neuron, right? So structural functional unit in the nervous system consists of cell body, dendrites, and axons. That's its anatomical composition. Functionally, neurons, there are three types of them: sensory, motor, and interneurons. So functionally speaking, that's how many types there are. For sure, you know that sensory neurons, they are receiving things. They're the ones which are kind of sensing the environment, right? Motor neurons are the ones which are actually acting, the ones which are innervating the muscle. There are two types of sensory: somatic afferent and visceral afferent. And I feel like this one gives nightmares to students. Somatic afferent, visceral afferent. What's the difference between them?
Somatic is the one you can actually feel, and visceral's like of the organs.
Very good. Yeah, so I wouldn't use the word feel. I wouldn't use that, because technically visceral could also be felt, but not in that area. I'll explain it right now. But yes, so visceral, most important, visceral is definitely organs. Okay, it's definitely organs. So what do I mean by organs? It could be glands of some sort, it could be blood vessels, it could be even mucous membranes. Most importantly, that visceral is autonomic. You're in no control of it whatsoever, right? Somatic afferent, it's the sensation of pain, temperature, touch, pressure, specifically from your body surface, but it could also be within your body as well, if you're talking about muscles, tendons, and joints. If I have to sum up what exactly somatic afferent neurons do, two words: pain and proprioception. That's what they do. Anybody knows what is proprioception?
Giving positional information.
Your position in the environment right now. So right now, if I close my eyes, very important that it's non-conscious sensation. I'm not aware of it, right? So if I close my eyes, I can tell you I am sitting on a chair, and I can tell you that my head is straight, right? It's straight. But if I close my eyes and I don't know, somebody rotates me, right? I will be able to still tell you which way are you rotating me. It's my non-conscious sensation, it's my non-conscious position in the environment. But how exactly am I aware of that? Well, of course, it's temperature, touch, pressure, and most importantly, my skin, my muscles, and my tendons. As they stretch, they give information to my brain in which position are they located. Like, I can tell you my hand is, I don't know, straight, or is it down, or is it flexed, or is it whatever. Okay, so I can tell you all of those things with my eyes closed. All of it is the job of the somatic afferent, right? So just pain and proprioception. For visceral afferent, we have said it's just your organs. Like, you're in no control whatsoever. Something happened to the organ and it's painful, probably, but does it mean that if I point that it hurts here, that the problem is here? Is that what it means? If you're talking about visceral afferent? Absolutely no, because last semester you should have learned that in the visceral pain situation, the transmission scatters, so it's not exact. In the somatic, if I say it hurts here, that means the problem is here. But in the visceral, because nerves innervate so many different organs, it does not mean that. Take a look at your appendix. Okay, so when you have the appendix pain, it's very rare that it actually hurts there. Most typically it hurts on the other side because it radiates towards that side, right? So that's your visceral afferent situation there. Okay, motor, somatic, efferent, visceral efferent, very easy. Somatic, voluntary, so that's your skeletal muscles. So visceral efferent, that's your smooth, cardiac, and of course, as always, glands. So what do you think interneurons do?
They would relay the sensation from like the sensory to the motor.
Yeah, they're the ones which are actually connecting sensory to motor. They're located between them. What do you think the job of interneurons is? Like in which particular functions it's the most important neuron? Sorry?
A reflex action.
Reflex. Every single reflex that you can find, interneuron has the most important job there, because it makes sure that why send all of this information to the brain and then wait until the brain decides what to do, right? So interneurons allows you to fully bypass the brain by simply connecting sensory to motor. So if once you touched the hot stove, which for sure we all did once in our life, you will never do that again, because your brain at first will forever associate the hot stuff with pain, thanks to the interneuron it has made. Fun fact, you know, when we learn, right now you're learning, right? So guess which neurons are we making? We're making interneurons. As you're writing right now, like whatever Yulia has said, you're writing, your brain associates that information. So when you will try to recall it, there's going to be an interneuron which connects this memory to you writing. So next time you write it, you will recall it. Okay, so that's interneurons right there for you. So this is why majority of neurons are interneurons. As they tell you, the more you read, the more synapses there are, right? The more you study, the more synapses there are. Yeah, but synapses between who and who? Synapses between interneurons and the rest of the neurons, like sensory and motor. So if you want to get smarter, you should make more interneurons, because they will be the ones. Okay, we talked about them. Learning, as I have said, they're super important, and decision making. We'll talk about it soon. Okay, so they are the ones which participate in that. There are two types of interneurons, local and relay. For sure, you can guess what's the difference between them. Local interneurons have a shorter axons, so as a result, they don't go to far distances, right? So they stay within local area, like in this part of the brain, and that's it. The relay interneurons have a much longer axon, so as a result, they can leave one part of the brain and go to completely the opposite part of the brain. Okay, so that's the most important part. Okay, most important part. Would you remind me, what are the functional types of neurons?
Somatic, huh? Somatic?
More or less, yeah. It's actually motor, sensory, and interneuron. Okay, so these are the different functional types of neurons: motor, sensory, and interneuron. What are the morphological, or what are the anatomical types of neurons? Like, how do they actually look like, right? So how do they actually shape like? We have 4 of them. We have unipolar, bipolar, multipolar, and pseudo-unipolar neurons. These are the 4 morphological, 4 anatomical types of neurons. The most common one, the one that which is also in your brain, is of course multipolar, because what do you see? How do we call this area?
Cell body.
Or
Soma.
or
Very.
pericardion. Very good. How do we call these?
Dendrites.
Dendrites and
Axon.
axon. So multipolar neuron. It has what? It has many dendrites and only 1 axon. Am I right? What do you think bipolar neuron? What does it have 2 of?
2 axons.
Yes, it has 2 axons. It has dendrites. You can see surprisingly dendrites in this neuron are attached to 1 axon. Then there is a cell body, and then there is another axon, right? Unipolar neuron. It also has 1 axon, but do you see it has 2 pairs of dendrites that do not come from the cell body, right? So they don't come from the cell body. So do you see the difference, bipolar and unipolar? Here we had 2 axons, out of which dendrites were coming in. Here we have 1 axon, out of which dendrites are coming in. And lastly, we have the pseudo-unipolar neuron. Pseudo-unipolar neuron, it's like a combination of 2, because you can see it does have like 1, I don't know, weird looking axon, and then this axon splits. It's like a combination of 2. There's nothing more that we can say. Like we had 1 axon and then it split into 2. So that's why it's pseudo-unipolar. But in reality, pseudo-unipolar technically is both. It's bipolar and unipolar. Like it has characteristics of both, of bipolar and unipolar. Yeah?
I have a question.
Yeah.
Is the multipolar neuron the only type that's myelinated?
Good question. It looks like it's the only one which is myelinated, but no, all of them are myelinated. Every single one of them. The artist was just too lazy to like.
You said the multipolar has many axons, right?
Multipolar has many dendrites, but 1 axon. Multipolar has many dendrites, but only 1 axon. Yeah. Bipolar has many dendrites, 2 axons. Unipolar has many dendrites, 1 axon, but you see that the dendrites are not coming from the cell body. They come from the axon. And pseudo-unipolar has dendrites, has axon, but they are not coming from the cell body. They come from this little foot process, right? So some kind of connection that you can see here. Okay, so now we need to combine the knowledge. Again, quickly, let's recall what are the functional types of the neurons?
Sensory, motor, and
Interneuron.
And inter. What are the anatomical or morphological types?
Multipolar, bipolar, pseudo-unipolar, and unipolar.
We need to combine the 2 pieces of information. So motor neurons and interneurons, the type of neurons they're multipolar. Forever and ever, you should remember this piece of information. Okay, so motor neurons and interneurons. Which kind of neurons are they? They're multipolar. So what does it mean? It means that they have 1 cell body, out of which we have many, many dendrites and only 1 axon as well, right? Sensory neurons are pseudo-unipolar. I apologize, I'm missing here. Okay, so they're pseudo-unipolar. Sensory neurons are pseudo-unipolar. Bipolar neurons are only present in 2 areas, retina of the eye and vestibular cochlear ganglion. That's the only location where you'll find bipolar. So which one are we missing? Which one we haven't talked about?
Unipolar.
Unipolar, very good. We haven't talked about unipolar. So just a quick recap. Give me an example of multipolar neuron.
Motor and interneuron.
Very good. Motor and interneurons are multipolar. Give me an example of pseudo-unipolar.
Sensory.
Sensory. Give me an example of bipolar.
In the retina and the
And-
ganglia of the vestibulocochlear nerve.
Exactly. So the one that we haven't talked about is unipolar. So here, please don't forget pseudo-unipolar. It's my fault. I need to change it, okay? Pseudo-unipolar. The sensory are pseudo-unipolar. So where in our body do we have unipolar neurons? Nowhere. The only place where we have unipolar neurons are in the insects. And there, unipolar neurons are responsible for glands, secretion, and contraction of the muscle. That's it. As a
Why are we
human,
studying it then?
sorry,
Why are we studying it then?
because it's neuro. We have to know that there are 4 types of them. Okay, like we have to know this. But yeah, okay. So insect. You will hear. By the way, most important teachers this semester, Professor Wolf, and that's it. Whatever Professor Wolf says, that's what you should go with, because he's the one which is the neuro person, okay? Not even Anton, not even Clara, Rosa, nobody, okay? So professor, they're also very good in neuro, but the one person is Wolf. So if you actually have him as your teacher, like honestly soak up every piece of detail that you can, because he's amazing at neuro, like he's so amazing. For neuroembryo, the best person, Wolf, and Super Mario. So surprisingly, that's his specialty. Okay, so Super Mario, it's the guy with the mustache. Okay, forgot his name. Laszlo? Laszlo? I don't know. I think Laszlo. Do you know who I'm talking about? No? Okay, that doesn't matter. But yeah, so these are the people that you should definitely hear. They will talk about that. Rosa, she says that unipolar and pseudo-unipolar are the same. Professor Wolf laughs. He says they're not the same. They're 2 different. Okay, so that's why, again, he's the one who'll be making the questions. So, okay, overview of sensory. You're gonna study this. Motor neurons. I want to add a bit of more details here. We have 2 types of motor neurons, 2 types of them, lower and upper motor neurons. Lower motor neurons are originating in the spinal cord, and they're the ones which are located in the spinal nerve, so they go for the innervation of the skeletal muscle. So lower motor neurons are between spinal cord and skeletal muscle, right? They're the ones which connect the 2. Upper motor neurons are between brain and spinal cord. Lower motor neurons have another name. They are also known as the anterior horn cell, or another name, alpha motor neuron. So 3 names, the same thing, okay? Lower motor neuron, alpha motor neuron, and anterior horn cell. Exactly the same thing. What are they between? Spinal cord and skeletal muscle. Upper motor neuron have another name. They are also known as the Betz cells. We'll talk about them later. Betz cells. So Betz cells are between brain and spinal cord. Okay, very important question. Everybody knows what is flaccid paralysis? Flaccid means that you're like, oh, like you can't move whatsoever. because there's another type, which is like jerky paralysis, where muscle does control, like it does contract randomly like this, okay? Like this, that's also going to be paralysis. But flaccid paralysis is when you're just like, just there's no movement whatsoever, nothing, okay? So zero. So flaccid paralysis will result if which of the neurons is damaged?
Motor.
Yes, but we have two of them, lower and upper. Which one?
Lower.
Lower. Very good. Lower. So because what is lower between?
Brain and spinal cord.
Spinal cord and skeletal muscle.
Spinal cord and skeletal muscle. So the one that is very important for contraction of the skeletal muscle. So if you actually damage the upper motor neuron, you can still have a bit of contraction. You can still feel just a bit of things, and you can still do some like reflexed contractions. There will still be there, but absolutely nothing, absolutely zero flaccid paralysis. That means that the lower motor neuron is damaged. The connection between spinal cord and skeletal muscle is lost. That's it, you're done. Okay? Okay. This is for you. You can read it at home. The most important thing is right here. They put it in the lecture, so I put it here, but like nobody cares. This is the question. What types of neurons are there in the brain? Isn't something we can fully answer yet. So all of this is for nothing. So here they're like debating, like, is it this neuron? Is it that neuron? Okay, so they cannot. Okay, so which kind of neurons are there in the brain is very difficult to say. So do you see what we have done right now? We have said that we have functional neurons like sensory, motor, inter. We have anatomical ones like, I don't know, pseudo, uni, bi, multipolar, and etc. We can actually classify them very nicely, right? In the brain, forget about it. Like, it's such a mess that you cannot classify anything. The only classification of neurons in the brain is actually based on the neurotransmitter that it is using. That's the only classification that we can kind of make sense of. The rest are impossible because it's like, it's a mess. It's like, it's completely a mess. Okay, movement. So you have said that this is the cell body of a neuron. We have a bunch of things there, bunch of organelles, blah, blah, blah. Could you tell me what is in the axon? Because yeah, for sure, in the cell body organelles, blah, blah, blah. But what's in the axon?
Microtubules.
Very good. You guys are so smart. Microtubules, yes. Anything else?
Microfilaments.
Sorry?
Microfilaments.
It's pretty much the same. So you could say that there are some filaments there, like actin filaments, yeah. Neurofilaments, yeah. Anything else?
Cytosol.
Cytosol, very good. How do we call cytosol in the axon? You're finishing second year, so for sure you can make up the word.
Exosome.
Axoplasm, because it's cytoplasm, okay, so axoplasm, okay, so, but axosol is also could be used, but yes, okay, so axoplasm, that's what you're gonna call it. So here we also have axoplasm, but for sure, yeah, the most important things that we have here are microtubules. Okay, could you tell me, what do microtubules consist of? That's histo 1 right there.
Tubulins.
Very good. Alpha and beta tubulins. That's the dimer that makes up the microtubule. So that's the microtubule. It has the units like dimers like this, right? So alpha, beta tubulins. Okay. If nucleus is here, so cell body is here, axonal terminal, this part, is here. Which charge is here?
Negative?
Negative. Why negative?
Because the other one's positive and I remember it that way.
Because the other one is positive, but come on, why is this one the negative one?
The DNA. is
DNA.
negative.
Say better. What exactly in the DNA is responsible for the negative charge?
The phosphate.
Phosphate.
Phosphate groups, very good. Because DNA is very rich in phosphates, and phosphates have a very negative charge. So as a result, the cell body, this area, it has a negative charge. The ends, they will have a positive charge simply because they don't have the phosphate groups. That's the answer, because they don't have it. Okay, so there is something which is walking on these microtubules, the motor proteins. How do we call them?
Kinesin and dyneins.
So which way is walking kinesin?
To the plus end.
To the plus end, very good. So kinesin is this way. So what is the charge of kinesin?
Negative.
Negative.
Negative, because it's walking away from the nucleus towards the end, so it has to have a negative charge. What about dynein?
It's positive because it's going to the negative charge.
Exactly. So it has to have a positive charge, so it's actually going towards the nucleus. So how do we call these movements? Do you see? One of them is going towards the cell body, one of them is going away. So anterograde or retrograde, that's the movement that you're talking about. So in the axon, you can find two types of flows, anterograde and retrograde. So which motor protein participates in the anterograde movement?
Dynein.
Anterograde is actually not towards the nucleus, it's actually towards the periphery. Yeah, and retrograde is towards the nucleus. Because think about it, here we have dendrites here like that, like we receive from here and we want to pass it all the way this way. That's the normal way of flow. So we would call it anterograde. So anterograde is from the nucleus all the way to the axonal end. So which one, I repeat, in the anterograde?
Kinesin.
Kinesin, because it's going towards this end. Dynein will participate in the retrograde flow. Could you tell me what are some of the things we could transport?
Neurotransmitters.
Very good. Neurotransmitters. Anything else?
Proteins.
Very good. Technically, you will see that neurotransmitter is a protein, but we could transfer some different proteins, sure. Anything else? Calcium, lipids, certain ions could be transferred there. Tubulins could be transferred there. Did I say lipids? Because lipids, ions of some sort, actin filaments could be transferred. Okay, and my favorite, that's the one that is my favorite. Agree with me that there's lots of energy needed. Like you're constantly walking, I don't know, you're doing things. So there are lots of energy that is needed to be, I don't know, in this area, for example. So guess what are they carrying like this?
ATP?
I wish. Mitochondria. The entire mitochondria can be carried all the way there. And it's like literally, that's how I imagine in my head at least. And I do apologize for such vivid picture that you're about to see, but I personally always imagine how kinesin is just walking with the mitochondria and just milking it like no tomorrow for the ATP production, you know, because there's so much ATP that has to be there. So why get like one little ATP, right? So why get one glass of milk when you can get the whole cow? Okay, so that's why exactly the kinesin just kind of stills the mitochondria and just sort of pushes it all the way this way, just carries it. So mitochondria could also be actually carried.
Would the dynein also carry the mitochondria or only the kinesin?
Very good question. So that's my next question. What will dynein carry? It's retrograde, it's going back. So what could it carry? It's very easy. Everything that hasn't been used, because recycling, guys, you know, so it has to be brought back to the cell body. Whatever your neuron did not use, so of course mitochondria is part of this list as well. So you're going to see that whatever you can use, whatever you did not use that could be used later will be brought back to the cell body by dying. Okay, we talked about cell body. A little bit more of a detail is that in neurohistology, we always see nucleolus, always. So this little tiny dot, this is not a nucleus, this is a nucleolus. Nucleus is this. Could you tell me what is the function of nucleolus?
Ribosomal RNA.
Amazing. That's the #1 place where ribosomes are actually being produced. So nucleolus contains lots and lots of RNA. So this should make sense to you. Why do we see nucleolus so clearly in the neuron?
Because it makes proteins.
Exactly. Because it's the most protein secreting cell there is. The amount of proteins that it makes is insane. So that's why it makes perfect sense that the amount of ribosomes and rough endoplasmic reticulum will also be super high. So high that we can actually see it in the cytoplasm and even has its own name. Nissl bodies, these are packed RER together with free ribosomes. Don't forget free ribosomes specifically. So these Nissl bodies that you see in the cytoplasm, they are super basophilic, right? Why are they basophilic?
Because proteins have a negative charge, they're made of protein, they make proteins.
So the things that make protein have a negative charge, but protein itself actually has a positive charge. Okay, so it's typically red. Think about collagen. Collagen is a glycoprotein, it's super red. Okay, so it's super eosinophilic. But yeah, you're right. So they are super basophilic because they are protein making organelles. So Nissl bodies, I repeat, packed rough endoplasmic reticulum with a bunch of free ribosomes. Why do we see the nucleolus? Because it is a protein secreting cell. Okay, back to axon hillock. Remember axon hillock? That's the area where axon starts. That's the area where initiation segment will be present. And also very important that this is the area that is not going to contain Golgi, ribosomes, and Nissl bodies. These things could be occasionally present in the dendrites. You could see these organelles in the dendrites, but in the axon hillock, absolutely not. Okay, we talked about this. So just a bit of a, like make sure that you get it. How do we call this entire thing?
Cell body.
This is the cell body of a neuron. Another name?
Soma.
Soma. Another name is perikaryon. Yeah.
Perikaryon.
How do we call this?
Nucleolus.
The whole thing, this is the nucleus, but this little tiny thing, this is going to be a nucleolus, right? What are these?
Nissl bodies.
Nissl bodies, and they are just a bunch of packed RER and free ribosomes, right? This is a very good slide because you can actually see the axon hillock very nicely, and you can see that it is much more paler than the rest of the structures, right? So definitely it gives you an idea that it's free of Nissl bodies, it's free of other things. And yeah. Okay, super important for the exam. If we were to compare dendrites and axons, like if I were to ask you right now, what do you think is bigger, dendrite or axon?
Axon.
And you would be
You would be
right because majority of the students, that's what they believe in, because that's what the pictures always show us. Do you see that whatever diagram we look at, always axon is so big and dendrites are just like little tiny branchy things. But in real life, take a look, that's the actual picture of a neuron. Take a look at axon. It's a little tiny thin structure that is going downwards. And take a look at the dendrites. It's insane. So actually, which one is bigger? Which one has a bigger diameter? It's actually going to be dendrites. Okay, so dendrites have a much more bigger diameter and axon is a bit thinner. Dendrites are unmyelinated, very important. Axon is myelinated. I know I say here Golgi is not found, and then here I go back and I'm saying that it is occasionally found. It depends, okay? So this is from the lectures. That one is from the book. So we have to check the lectures this year. What do they say? Do we find Golgi in dendrites or not? Okay. The rest we have talked about it, but yeah. If you need help to identify dendrites or axon, like whatever you're looking at, that's the one that I'm always using. First of all, axon will never have branches. You will never see little tiny thing coming out from the axon. Okay, it's a kind of uniform looking straight line. Another thing for axon is that its thickness is consistent throughout. So if I start here, this thickness is the same as this thickness, the same as this thickness. Take a look at dendrites. It starts super thick, but then it becomes thinner and thinner and thinner, like a typical tree situation, right? So the branches of a tree, the trunk is thick, and then as you move a bit upwards, it's going to be definitely thin. And of course, dendrites do branch out. You can see that they do have branching. Okay, synapses, we have talked about them. So they are communication between the neurons. The different types are axodendritic, axosomatic, axonic, and don't forget the one more is going to be the dendrodendritic, which is only in theory. In real life, we couldn't find a single one, right? So there is no dendrodendritic in real life. Could you give me one more? So axodendritic, axosomatic, axonic, dendrodendritic, and last one that we have in our body.
Dendrosomatic?
Dendrosomatic. Nope, because dendrites are kind of coming out from the soma, so that's not the synapse, it's like just their connection.
Dendroaxonic?
Somatosomatic?
Nope. Somatic, nope. Dendroaxonic, you're switching them. So you're saying that dendrites are transmitting and axon is receiving. So it's not how it is, right? Come on, you know it. Sorry?
I got confused. Can you just say it?
Yeah, so if you say dendroaxonic, you're switching them. Here, which one is going to be like one of them is receiving and one of them is transmitting, right? So if you're saying dendroaxonic, so definitely dendroaxonic is a good one, but one of them is receiving, one of them is transmitting. Like there's nothing more that you can say. Just trust me, it's not there. So axodendritic, that's the one that it should go because it starts to transmit. Do you know what I mean? Like you don't receive first, you first transmit. It's like a situation with chicken and the egg. What is first? Okay, so the same thing is here. What do you start first, transmitting or receiving? You start transmitting first. So axo has to be first. Axo is always first because it starts to transmit things. Do you get it? So one more. You know it. Axo something. Axoeffector. A very good example is neuromuscular junction. It's between an axon and a muscle, skeletal muscle to be more specific. So axonoeffectoral, sorry. Okay, so axonoeffector synapse. So it's between an axon and effector structure. Can we see synapses in H and E? No, you need silver precipitation. By the way, silver precipitation, silver impregnation in neurohistology has a different name. It's actually called Golgi method. So make sure you remember this. We'll have a couple of slides. So silver precipitation is going to be Golgi. Synapses functionally are classified as chemical or electrical. Electrical is, you know, is the free flow of ions, gap junctions, etc. Where do we have electrical synapse?
And muscles?
Where specifically?
The heart.
In the cardiac, yes. And smooth muscle is also capable of having electrical as well, because they also have a bunch of gap junctions, but mostly cardiac, of course. Most important, the one which is most common, is definitely the chemical synapse, because that's where we use the neurotransmitters. So let's do this. The whole steps, chemical synapse. So how do we call this part?
Presynaptic.
Presynaptic neuron.
Presynaptic, which part of a neuron?
Axon terminal?
Axon.
Terminal, very good. It's a presynaptic axon. This is a postsynaptic dendrite. So that brings us to the same thing. Transmitting, receiving. Never receiving, transmitting. Okay, always you first start with the transmitting and then the receiving part. How do we call the gap?
Synaptic cleft.
Very good. Synaptic cleft. Okay, let's imagine. Never mind. What's the first step?
Action potential.
Very good. Before that.
So neurotransmitters are carried to the terminal.
Very good. But before that, you first make the neurotransmitter. Don't forget about that, okay? In the oral, exactly, in the physio, if you're going to be talking about this, if you get this topic in physiology oral, the first step is to make the neurotransmitter, because without that neurotransmitter, nothing is going to be happening. So once you made the neurotransmitter, very good, Ryota. The second is that you're going to be bringing that neurotransmitter all the way to this terminal end. How do we call this flow?
Axonal transport?
Yes, but in that particular direction.
Anterolateral.
Anterograde.
Anterograde
Flow, right? So it's going to be through the anterograde flow, the neurotransmitter is going to be brought up to the terminal end. Let's imagine that this neurotransmitter is acetylcholine. We're going to be talking about acetylcholine as the neurotransmitter. What is the next step? Anuli has said that action potential starts to travel, right? So it travels along the plasma membrane. What will that do?
Opens calcium channels.
Why are they opening up?
They are voltage gated.
So what do they sense?
Action potential.
Say better.
Depolarization.
Change in action potential.
Say better, you're almost there. Change in
Resting
potential.
in brain potential.
change in polarity, because you know that action potential, for example, the plasma membrane was negative, right? So it was negative, but as depolarization happens, it becomes positive. So change in the charge, change in the polarity. So voltage gated calcium channels, they sense it, so they open up. So what will that do? Of course.
Allow fusion of vesicles containing
So calcium
neurotransmitters.
goes in, yeah. And what will calcium do?
release the neurotransmitter.
How exactly?
By fusion of the vesicles onto
How
the
exactly do they fuse?
with the help of
Something like
snare
this.
proteins.
There you go. The key situation that you need to talk about are the snare complex. So snare complex, you know, we have the T snare, right? So the terminal one, we have the V snare. So this one is the T snare, this one is the V snare. So one is on the vesicle, one is on the terminal plasma membrane somewhere here. Thanks to calcium, without this calcium, the snare proteins will not work together. So that's why we need the influx of calcium. So once the calcium is here, once the two snare proteins form a complex, the plasma membrane of the vesicle fuses with the plasma membrane of the neuron, releasing the neurotransmitter into the synaptic cleft. What's then?
Diffusion.
So it diffuses to the postsynaptic area. It is an acetylcholine. Let's say it is a muscarinic one. So what does it mean?
It's a G protein.
So acetylcholine is going to bind to a G protein sodium receptor, right? So sodium is going to be going in. Let's say it is a nicotinic.
The ion channel.
Then it's just
Ion channel,
normal.
very good. So it simply binds to the this acetylcholine will simply bind to the ion channel. Ion channels will go in. Either way, in both muscarinic and nicotinic, the end result is that ions are going in. The ions will be flowing in a different manner, but they will be flowing in, right? These ions flow in. What do they do to this?
Depolarize.
Depolarize the membrane.
Depolarization. So action potential begins. It's going to start flowing in the other way. Overview of what we have talked about is here, but I strongly recommend you refresh your memory here. Okay, so a bit more, especially when it comes to snare. Okay, speaking of neurotransmitters, we have two different groups of neurotransmitters. We have the excitatory and we have the inhibitory neurotransmitters. An example of excitatory neurotransmitters are acetylcholine, glutamine, and serotonin. These are excitatory neurotransmitters. How do they work? Well, they have to cause depolarization. So what should they do? They should open a cation channel, most commonly sodium. That's what we can see here, right? So you can see that sodium channels are opening up. So it's an excitatory, it causes depolarization. Inhibitory is going to be the GABA and glycine. These are the neurotransmitters which are inhibitory. GABA stands for gamma aminobutyric acid, right? So GABA or glycine. They are inhibitory, so they don't want to depolarize the neuron. On the opposite, they want to hyper or repolarize it. So make it even more negative, so it's much more difficult to generate another action potential. So definitely they should not open cation, they should open an anion channel. Most commonly is going to be chloride. So for sure, whenever you're thinking about excitatory neurotransmitters, sodium channels, most commonly. Inhibitory, chlorine. So they are neurotransmitters, so on which receptors are they working? I strongly recommend you go over it. For your physio and for your neuro, it's very important to know this. Muscarinic and nicotinic. So you know the two types for the acetylcholine. Catecholamines, so norepi, epi, and dopamine. Okay, so these are the catecholamines. Their receptors, we call it adrenergic. Could you give me an examples of adrenergic receptors?
Beta and alpha.
Very good. So could you name me all of them? How many do we have alpha? How many do we have beta?
Alpha 2 and 3 betas.
Very good. Alpha 1, alpha 2, beta 1, beta 2, beta 3. You see there are 3 neurotransmitters. Which adrenergic receptor has the highest affinity for epinephrine specifically? There are 5 of them. Alpha 1, alpha 2, beta 1,
Beta receptors.
Beta 1. Beta 1.
2. Beta 2. Beta 1 has the same affinity for epinephrine and norepinephrine. They're equal. Epi and norepi equal affinity for beta 1. But beta 2 has the highest affinity for epinephrine. So whenever you have epinephrine, it goes specifically for the B2. That's where it goes. So homework for you, check where in our body do we have B2 receptors. So if you have high epinephrine, where will the B2 be present, right? So where will the epinephrine go? The rest of them have a very high affinity for norepinephrine. For sure, you know that norepi is your resting one, and epi is when you're like stressed. Okay, so yeah. Serotonin. Its other name is 5-hydroxytryptamine. This is why if you see 5-HT, do not get confused. 5-HT is just another name for serotonin, 5-hydroxytryptamine. This is a neurotransmitter in the central nervous system. So we will see it a lot there, but not only, also enteric. Your GI works on serotonin. So that's why it's very important, I think, to mention it here, where majority of serotonin is being produced in our body. Where do we make most of serotonin?
The gastrointestinal tract?
Exactly. That's the #1 place where you make serotonin. So all of those dietitians who say you are what you eat, they're absolutely right. The production of serotonin begins on what do you eat. Okay, so it's definitely that. And this serotonin will be used not only by your GI, but also by your brain, because your brain does make serotonin, but not as much. Okay, and I'm gonna stop here. So this is just for you, right? So what different neurotransmitters are doing. So epinephrine, you know, fight or flight. Norepinephrine, it's a calming one, the concentration. So when you're at your just resting self, when you're concentrated, norepi is there. Dopamine, you know, happiness, pleasure. Serotonin, mood, you are what you eat. GABA is calming, acetylcholine is learning. Right now your ACH is like through the roof. Glutamate, memory, and endorphins are euphoria. So these ones are also the happiness. Okay, and this is where we're going to stop. So that's it for today. Okay, guys, so that's it for today. Stop the recording.